I'm Amy Turner, and today I'll be presenting on pharmacogenomic genotyping performance across biological specimens. We'll start with a little bit of background on preemptive genotyping. Preemptive genotyping of relevant pharmacogenetic genes and HLA typing for association with the ADME and hypersensitivity enables a clinician to provide a patient with an optimized treatment regimen by maximizing both drug efficacy and limiting adverse reactions. Currently, there are over 300 actionable genetic variants with dosing guidelines on FDA-approved medications. These drugs span a wide range of categories, including pain management and cancer, and they impact a significant percentage of prescription medications. As shown in Figure 1 from the Relling and Evans Nature paper, that there were in 2015, 7% of FDA-approved medications contained at least one actionable pharmacogenetic relevant gene. The prescriptions in the United States impacted by this were 18% of total prescriptions, and this was 18% of 4 billion prescriptions. So this was a significant number of prescriptions that would potentially be impacted by actionable pharmacogenetic genes. Additionally, there are high-risk genotypes, and these vary in frequency between different ethnicities and populations, with certain high-risk alleles being common in some populations and not as common in others. So as shown in Figure 2, from the 2015 Dunneberger et al. Paper, you can see comparing Caucasian and African Americans. And when you look at the percentage of the population that carries at least one high impact allele for each of these genes, you can see in genes like CYP3A5, 69% of African Americans carry one of those, whereas only 15% of Caucasians do. However, if you look at CYP2C9, it's kind of the inverse of that, is 36% of Caucasians carry a high risk allele and only 6% of African Americans. So results like these really highlight that, one, looking at Figure 1, there's a significant number of prescriptions that are prescribed annually that could be impacted by pharmacogenetics. And that, two, there are different frequencies of these SNPs between different populations, really highlighting the need for comprehensiveness as well as an understanding of the risk factors for each particular patient population. The methods used in this study today include genomic DNA that was acquired from the Coriell Institute, or genomic DNA that was isolated in-house either from liver, blood, buccal, or saliva. Samples were batched, and this included mixing sample types per plate, and they were run on either the Pharmacoscan assay kit 24 format or the Pharmacofocus assay kit 96 format. The array analysis was done on the Axiom analysis suite, and performance of the assay was determined looking at both inter and intra-run specificity, sensitivity, and concordance. Table 1 highlights a subset of the genes that are included on both platforms. This includes the comprehensive list of CPIC genes with actual guidelines, a larger set of haplotype calling genes kind of pulled from CPIC and from GKB that have literature that indicates that there are actionable steps that could be taken, however, there aren't current guidelines. And then there are also 5 genes that had copy number analysis. The figures of the arrays just kind of show the different formats. You can have a 96 format, you can have a 384 format, or you can have a 24 format with the larger array surface. The sample information that was included in the study includes information on Pharmacoscan samples, which were 112 Coriell samples, 161 liver samples, 20 blood, and 20 saliva. And the blood and saliva were matched sets and acquired from the same donors. Analysis was done on Axiom Analysis Suite 4.1. And additionally, the blood, saliva, and Coriell samples were also run on the DMET array and received CYP2D6 TACMAN-based copy number analysis. The samples that were run on Pharmacofocus included 127 Coriell samples, 27 blood, 27 saliva, also matched, and then 4 buccal samples. The buccal samples also had matched saliva results. Analysis was done on the Axiom Analysis Suite 5.0, and additionally, all of the samples included on the Pharmacofocus run were also run on Pharmacoscan or DMET array. The results for Pharmacoscan showed that all of the samples interrogated had a passing call rate of greater than 98% for all array quality control metrics. Genomic DNA samples from blood, saliva, and Coriell demonstrated a concordance range of, a QC call rate range of 98.85 to 99.99%. And then they showed inter and inter-run concordance rates that were greater than 98% for both plates. The liver-derived genomic DNA samples had call rates of 99.67 to 99.93%, and the diagnostic sensitivity and specificity were determined and shown in tables 3 and 4. Overall, all samples looked at and analyzed on Pharmacoscan showed high concordance with both known genotypes and with inter and intra-run genotyping performance. The results for Pharmacofocus also showed high levels of concordance and accuracy and specificity and sensitivity. All of the samples interrogated had a greater than 98% for array quality control metrics. And looking at plate 1 and 2, 91 unique genomic DNA samples from Coriell and 2 kit supply control samples were analyzed. Concordance between plates 1 and plate 2, as well as concordance between PharmacoScan results and Pharmacofocus results, are shown in figure 3. On average, the concordance results were greater than 99.5% looking at all combinations of analysis. So Pharmacofocus plate 1 versus plate 2, and then Pharmacofocus plate 1 and plate 2 versus Pharmacoscan plates 1 and plate 2. This was a high level of concordance, showing that the reliability and reproducibility of both of the assays. For plate 3 analysis, 34 genomic DNA samples from Coriell, 4 buccal samples, 27 blood, 29 saliva, and 2 kit supply control samples were analyzed. Buccal samples were collected using 2 different extraction kits, Genturi and Isohelix, and samples from the same donor were also collected from extracted saliva. Concordance results were shown in table 5. And overall, we had a great concordance between all the different sample methods, as well as passing QC call rates for all of the different collection kits. Looking at the different sample types, it can be very important because different groups of people can have different troubles collecting different ways. For example, some medications lead to dry mouth, so saliva sample might not be good for collecting for someone on certain medications, whereas a buccal might be. However, in general, buccals often yield less DNA or might have different quality metrics than saliva or blood. So this really highlights the difference that you can see between sample types, and it also highlights the need to validate and interrogate your assays using multiple sample types. Overall, regardless of the DNA origin, samples produce results that met array requirements for both assays. Comparison with 1,000 genomes data for the Coriell samples and then DMET results allowed for comparison of the determination of accuracy of blood, saliva, and Coriell samples on Pharmacoscan, as well as Pharmacofocus. And of note, the array accuracy detected copy number variation for CYP2D6, as well as hybrid or gene conversion arrangements. And it detected accurately on both platforms, and this is integral for metabolizer status prediction for this gene. And up to 20% of people do carry a copy number or structural variation for CYP2D6. Downstream inclusion of this data into the EMR of the patients will enable clinicians to preemptively make decisions for drug choices, dosing decisions, and hopefully provide a cost-effective and better individualized patient care. Thank you very much for your time on this, and these are just the references that were included in this poster. Thank you.